Well, good afternoon. I would like to thank the chapter and the organizers for the invitation. It's a pleasure to be here. And what I would like to do is take some of the concepts we discussed today, put that slightly in different context, and tell you about our experience and our goals of taking really comprehensive biological big data, apply very sophisticated algorithms that were just described, and show you how we're thinking about translating that into improving the process of drug development, improving the process of biomarker development, and how ultimately we believe that will actually change clinical care. So the key concept that I will be focusing on today is this concept of multi-omics, or as we refer to as Panomics. And just to define that phrase, we mean by Panomics is the application of technologies like genomics, transcriptomics, proteomics, all together, and apply that to the drug development and biomarker development process. And that's what I would like to talk to you about today. So, two main points that I would like to get across. First is that, as it has been said before, is that the balance between health and disease is really determined in the context of the environmental influences, in the context of the DNA, RNA, protein, small molecule continuum. So it just follows that following this paradigm is really the logical way to do discovery today. And secondly, we believe that this Panomics-based biological big data, when it's coupled with precise quantitative phenotype measurements and then leveraging artificial intelligence-based tools, is in fact what is going to change, number one, the way we discover and develop drugs, secondly, the way we discover and develop new biomarkers, and eventually how we practice medicine. So to be a little futuristic, I published this paper a number of years ago. So I would like to start out with a case presentation. So imagine that you have a 20-year-old female who at 16 weeks of pregnancy comes for a routine visit, has a blood test at the physician's office, and now from the blood we perform whole genome sequencing of the fetus, which is a technology which is present and possible today. A number of weeks later, a healthy baby is born. And as we continue with the case presentation, by the time this fellow is 18 years old, because of a gain of function variant in the PCSK9 gene, they received the PCSK9 vaccine. Because of some of the genetic susceptibilities that were identified at the time of the fetal whole genome sequencing, they are monitored throughout life for prostate-related problems and eye-related problems. At age 40, all these issues turn out just fine. Now at age 43, this person now just has gotten through a upper respiratory infection, and when he goes to the restroom about 30 minutes after dinner, the automated toilet sensor indicates that there's an elevated blood sugar now in the urine. So then, of course, an insulin resistance diabetes panel is ran through the phone, which is then transmitted to the hospital. And so the diagnosis is now made of viral-induced insulin resistance on the background of genetic susceptibility. Now, whole panomic profile is performed, and on the basis of the proteomic and the transcriptomic data, now there's a personalized treatment which is applied, and the insulin resistance is reversed. There's no atherosclerosis on the imaging at this time. At age 56, because of the genetic susceptibility that was identified 56 years prior based on whole genome sequencing, now a targeted imaging procedure identifies a mass in the prostate, which is now with a targeted delivery system. A drug is now delivered to the endothelial epithelial cells of the prostate, and this completely resolved. In 2085, at age 72, the mirror at home identifies some problems with the retina. So obviously the diagnosis of macular degeneration is made. The retina is regrown in the lab, is reimplanted, at which time the patient's pilot's license is reinstated, and in 2106, at age 93, the patient remarries and lives another 19 years. So this is sort of how we believe that digital medicine will in fact change how actually we apply this in the clinical practice. So question is, how do we get there? So our approach has been for the past 5 years is to take a real comprehensive biological view of how disease develops. And the concept is really very simple, and my 8-year-old learned this in biology over the past several months now, is that, of course, disease develops in the context of environmental influences along the DNA, RNA, protein, small molecule continuum. So the thought we had about 5 years ago was that what if we collected one large patient population that was large enough to do the proper, appropriate statistical, genetic, and other analyses, if in this large patient population we actually measured disease quantitatively very precisely using imaging methodologies, and then simultaneously measured the DNA, the RNA, the protein, effectively every single molecule we can think of in the human body based on the technologies that are available today. So the fundamental concept has been really very simple. First is to use imaging to really differentiate cases from controls, those with a disease versus without a particular disease, in this case atherosclerosis. And then once we separate the cases from controls, which is the task for imaging, also to make very specific quantitative measurements of a lot of the traits that are relevant to that disease. What kind of plaque do you have? How much of plaque do you have? Where in the arteries is that plaque? So that was really the idea behind the company about 5 years ago. The good news is that this concept is not only applicable to cardiovascular disease, but to a host of other conditions. So in this context, we are looking at things like atherosclerosis, aortic valve disease, mitral valve disease, fatty liver disease, bone disease, effectively all these different conditions where we can get a quantitative phenotypic measurement of the diseases we're interested in. Now, we were very focused on quality and execution here, so we did not go to existing biobanks, we did not go to academic consortia. Instead, we were fortunate enough to secure funding to actually do a very large prospective clinical trial in order to gather this data. So we designed our study, which was called the Global Clinical Study, which recruited prospectively nearly 8,000 patients. We designed the study on the basis of the Institute of Medicine guidelines and the AHA guidelines for biomarker discovery, with very specifically pre-designed analysis cohorts that were determined a priori prior to enrolling the patients. We then completed the enrollment of nearly 8,000 patients in just 17 months at 48 clinical sites, 9 countries in 3 continents, with a small 15-member biotech startup company, which was quite the achievement for us. But conceptually, there are 3 components that are relevant in terms of the approach. The first pillar is, as I said, the phenotyping component, which is absolutely critical. That is, we measure disease quantitatively. The second pillar is the panomic approach. And then third, of course, is applying these machine-based learning algorithms to then take this raw data, trillions of data points, and identify the relevant biologies which we can then translate into drug development and biomarker development. So a few words about each of those components. So the phenotyping aspect was my personal background. I trained in non-invasive imaging, including cardiac MR and cardiac CT. And I spent about 12 years of my career prior to starting G3 on really identifying and positioning cardiac CT as a non-invasive phenotyping tool for atherosclerosis. We published a number of papers, including one in Nature, showing how we actually utilize cardiac CT for phenotyping, and we validated that against the entire spectrum of modalities, including histopathology, invasive angiography, intravascular ultrasound, near-infrared spectroscopy, and even fractional flow reserve. So we spent a lot of time figuring out how to quantitatively measure the phenotype traits that we're interested in. So when we performed our study, we actually collected these 8,000 CTs. We trained each of the sites. We have the images. Our core lab is analyzing the images, and we're also using artificial intelligence-based imaging approaches to actually develop these automated tools against the reads of the experts in the core lab, which is we have the largest data set for this in cardiac CT. Our core lab makes 450 quantitative measurements of the coronary arteries in each patient. So we'll look at things like plaque type, plaque features, plaque pattern, the calcification type. We of course look at the degree of the stenosis, and we look at very specific high-risk features such as the napkin ring signs and things like that, which are clearly the most vulnerable phenotypes. But again, what's critical here is the application of very specific quantitative measurements of these phenotypes. And in fact, this observation, which we published a number of years later, which is shown on the screen now, was the very observation that prompted me to start this company 5 years ago. And this was when we were working on validating cardiac CT against a number of different modalities, and we made the observation that when we use cardiac CT to separate cases from controls for the presence or absence of atherosclerosis, we reclassified 30% of the patients relative to case control status compared to invasive angiography. Now, when you do omics-based big data discovery, and you're literally comparing 28,000 genes in cases versus controls, separating cases from controls is an absolute prerequisite. And the better you separate them, the higher the signal-to-noise ratio, the more the signal, the less the noise. So this was the original idea behind actually taking CT as the phenotyping tool and couple that with Panomics. But again, we're not only applying this for cardiovascular disease, but also for fatty liver disease, a major medical unmet need today. And we're again able to quantitatively quantify fat content in the liver, fibrosis content in the liver in a continuous scale, which is key to these discovery efforts. We do the same to osteoporosis, etc. So phenotyping, quantitative precise phenotyping is the first major aspect. The second aspect of what we do, of course, is what we refer to as next generation Panomics, and perhaps this is probably the most unique feature of what we had done. So Panomics refers to the idea of taking all these omics technologies and measurements and doing them simultaneously. So what is it that we're actually performing in these patients? First, we do whole genome sequencing. This is not genotyping, not exome sequencing, it's whole genome sequencing, all 3 billion nucleotide in every single patient. That's 1 terabyte of data per patient just on the whole genomes. Then we do DNA methylation analysis, RNA sequencing, proteomics, metabolomics, lipidomics, and glycomics, and a lot of other biomarkers. This is truly big data. This generates 22 trillion with a T raw data points that we're then assembling in a completely unbiased fashion, utilizing these artificial intelligence-based methodologies. So how we do that is obviously beyond the scope for this discussion this afternoon, but we have developed a very systematic step-by-step approach as to how we apply, how we analyze this data. So in the next few minutes, I want to spend a few minutes on giving you some really high-level insights into some of the scientific observations that we've had to date, sitting on this really rich, very comprehensive data set. So again, we apply increasingly more complex analytics tools to this data set, starting with very simple hierarchical clustering, which is a typical way of looking at big data sets. Once we measure hundreds of thousands of traits or molecular measurements, and we start to correlate those to the phenotype, then the first thing to ask is, what are the big structures in the data set? What are the big clusters? What are the big groups of molecules that actually make a difference? Then, of course, we go into more specific approaches and ask the question of, now we measure 2,000 proteins in this patient and quantify 2,000 proteins. Which are the top 10 that provide some sort of incremental diagnostic value? We really utilize that to develop our diagnostic biomarkers, which are now approaching commercialization. Unfortunately, I was not able to pull up my PowerPoint. This is a PDF slide, so this was supposed to build. But what the slide shows, this is a map of the human metabolic pathways. And we measured about 1,100 small molecules from the circulation using mass spectrometry, and we were able to then identify metabolites that are upregulated versus downregulated in patients, in this case with cardiovascular disease. And what you can see is that there are very specific patterns that emerge, and there's very specific biochemical pathways that are dysregulated simultaneously in patients with atherosclerosis. So this provides incredible biological insight for us. This gives us the opportunity to develop our diagnostic algorithms, and this also gives us the opportunity to identify pathways and enzymes in the pathways which then become potential drug targets. Because when we apply our multivariable data sets, multivariable analysis tools to the data set, what we find is that we identify key molecules from each of the biochemical pathways that represent the abnormality in that particular pathway. We take those analytes and build them into an algorithm, and now we have a diagnostic signature for cardiovascular disease, one for fatty liver disease, one for osteoporosis, all on the basis of this completely unbiased analysis of the big data. Then one of the very amazing observations we had very early on is when we started to look at this data set, is that the insights gained from these different omics data sets, which were, by the way, done by completely different labs, completely independent and blinded of each other, and yet when we looked at the top hits in the different omics, proteomics and metabolomics and lipidomics, actually the top hits for each disease state converged down to the same biological pathway. So here's an example where metabolomics, proteomics, lipidomics, gene expression data all pointed to a very specific pathway, which now we believe to be incredibly important in atherosclerosis. And this enzyme in that particular pathway is of great interest to us because we believe that that has a major role in the initiation of atherosclerosis. And of course, that's one of our major drug targets today. One of the key aspects here that we're trying to really emphasize is that we're taking a very unbiased approach to analyzing the data. So one of the most helpful platforms and tools we had found is Bayesian network analysis, but we use a proprietary implementation of Bayesian nets. And this is a completely unbiased way of looking at these data points and assembling causal biological pathways. At the key word being causal, because obviously we're doing drug development. And so I call this my $30 million slide because it took me 5 years and $30 million to get to this slide, which was our first pre-specified analysis point where we had about 700 patients and we had every single data point available. Whole genome sequencing, DNA methylation, microRNAs, messenger RNAs, etc. And in a completely, there was 1 trillion raw data points that went into this analysis. And we just, as a very simple question, we said, what are the biological networks that underlie atherosclerotic cardiovascular disease? And the most important, lots of important things to say about this slide. One of them is in this completely unbiased fashion, out of 1 trillion raw data points, we, quote, rediscovered the LDL pathway, including things like LDL, ApoB, Lp(a), PCSK9, etc. It's huge. If we didn't know about PCSK9 as a target, we would have discovered it in this context. And of course, we identified several novel pathways not intersecting with the LDL pathway, which are of great interest to us, and we're able to use that for very specific mechanistic insights. So that's the science. Now, one of our 2 commercial main goals is to then apply this for drug development and biomarker development, so let me show you a little bit into how we do that. So Raj actually, I think, showed this morning, which referred to Moore's law, which the general principle of the exponentially increasing computing power over the past half decade or half now century that has really transformed technologies. I don't know if you know this, but there's a concept in drug development called Eroom's law. Eroom actually is Moore spelled backwards. And what this shows on this graph is that the number of new drugs coming to the market per $1 billion spent on R&D has been decreasing exponentially, speaking to the inefficiency of our drug development process. So the question is, how do we change that? And one of the ways we change that is applying this Panomic big data, and we at G3 anchor that in the concept of genetic target validation. I don't have time to go into the details of genetic target validation, but the idea goes something like this. Since we sequenced every single gene in the human genome in thousands of people, we measured hundreds of thousands of intermediate biomarkers, and we have the phenotype measurements. We're able to close the loop where we first identify biomarkers that are associated with the disease. Example would be LDL cholesterol is associated with cardiovascular disease. Then secondly, we go back in the genome and identify variants that cause lifetime high or lifetime low levels of that biomarker. Example would be PCSK9 loss of function variants are associated with lifetime low levels of LDL cholesterol. And step 3, we then look at those variants against the disease, and if there's an association, then we close the loop where LDL is now causal in atherosclerosis and PCSK9 is a genetically validated target. Now, since we've sequenced every single gene in the human genome, we actually theoretically can do genetic target validation for every single gene or every single target that could become a potential drug target. I like to show this slide to show the validation of our platform. The image that you see on the left was published in 2010 in Nature Drug Discovery, showing the very well-known sigmoid relationship between LDL cholesterol and cardiovascular disease with the superimposed genetic variants. Fast forward 5 years, and I had that image, by the way, in my pitch deck when I was looking for funding for G3 5 years ago. Fast forward 4 years or 5 years, the image on the right is from our data set. when we actually plotted that LDL cardiovascular relationship with the PCSK9 variants, as you can see, our data set exactly reproduces the known biology. And there's a lot of examples of this, but it's very important for us to show that, in fact, our data set is very valid and very appropriate to make these discoveries. We then, of course, take these approaches and, again, apply them to the drug development process. And I mentioned Mendelian randomization being an important tool for us, and we came up with some interesting improvements on the Mendelian randomization process, where we are now able to take two target genes simultaneously. In this case, we took HMG-CR, which is the target of statins, and PCSK9, which is kind of a new drug being evaluated, and we performed the so-called 2x2 factorial Mendelian randomization study, just like you would do in a real clinical trial. And we were able to predict the outcome of the ongoing phase 3 clinical trials of PCSK9 inhibition on top of a statin. So we put our stake in the sand back in December when we published this in the New England Journal, and we said, well, we believe based on the genetic data that the primary endpoint will be reduced by 20 to 25%. In fact, 3 months later, when the study 4-year was presented at ACC, of course, they found the 20% reduction in the primary endpoint. So clearly you can use these genetic methodologies now to predict the outcome of these phase 3 clinical trials. So that's the ultimate validation of a drug target. So the progression of what we do now for genetic target identification and validation goes like this. We have a publicly announced collaboration with Sanofi in their PCSK9 program. So in that, out of the trillion raw data points that I mentioned to you, they asked us to look at a very specific pathway, and we identified a very specific new target from the Bayesian network analysis, from the gene expression data, suggesting one particular receptor to be very important for one particular lipoprotein metabolism. We then took that insight from the gene expression data that came out of the functional gene expression networks, and we're able to validate against the DNA, the gene at the DNA level of that particular receptor. So now we deliver this target to Sanofi, and Sanofi is in the process of finishing up the in vitro validation studies, and by July we should have in vitro validation whether or not this target actually could be translatable for a new drug. We can take the same concept, and rather than in this 2x2 Mendelian randomization approach simulate two targets of therapy, we can take one gene to simulate a biomarker as a companion diagnostic, the other one to simulate the target, and we can create a biomarker target pair, and we can show, again, using the genetic data, that we are able to show efficacy of that drug in a patient population with the abnormal level of that biomarker. That's the ultimate precision medicine implementation of these approaches. And we can now then use that to actually repurpose existing drugs. We just created a repurposing engine recently, which utilizes this exact methodology. So in that context, our value proposition for the drug development process goes something like this. Here's a target. Here's the mechanism of action and the pathway. Here's the predicted side effects. Here's a companion diagnostic that goes with that. Here's how we should design the phase 3 clinical trial. And by the way, this is what it's going to show. And we know this before we inject the first mouse with the first dose of the experimental drug. And that's what changes the paradigm. So if you look at the traditional drug development process today, it takes 24 drug programs to be funded and developed to take one drug to the market. This is why it costs $2.6 billion to get one drug to the market. We've modeled that with our approach, you change that to about a 7-to-1 ratio, major portfolio optimization. It's a 3.5x increase in efficiency, and so now it takes less than $1 billion to take a drug to the market, and that's our value proposition. And finally, in the last few minutes, I wanted to show you how we can alter the process of the biomarker development and discovery process as well. Our lead product is a product that we're calling No Plaque. The idea being that you do the test, and if the test is negative, then the patient has no plaque. So we are looking at the unmet need are those patients who present to the outpatient setting in the absence of a history of cardiovascular and coronary disease with chest pain. The unmet need, as you know, is that evaluation pathway today is quite inefficient. So the value proposition is that if we had a blood test which could act as a gatekeeper to the current evaluation standard, that would be an important value proposition. What are we supposed to do with chest pain patients? We'd have to do a clinical risk assessment. And then if it's an intermediate likelihood patient, we send them to the stress lab. If the stress test is positive, they go to the cath lab. The problem, as you know, in the United States, of the nuclear stress tests performed, I don't know if you know this, but only 5% of the stress tests performed in the US annually have actually ischemia on them. And then you take the 5% with ischemia to the cath lab, less than 40% will have obstructive disease. So that's kind of the inefficient process we're trying to address by introducing a gatekeeper blood test, no plaque. Idea being you have chest pain, you come to the office, we do the test, we calibrate it to have high negative predictive value. If the test is negative, then there's no need for further evaluation. If the test is positive, then you go into the non-invasive workup. We've gone through the scientific discovery and the validation of this algorithm. That paper is in the submission process right now. And in this context, out of about 1,200 different small molecules we measured in mass spectrometry, we identified 8 small molecules that are combined into an algorithm, and that algorithm then has a very high negative predictive value in the context of patients with chest pain, has an area under the curve over 0.8, and put that into context that is much higher compared to, for example, nuclear stress testing, and that is much higher relative to some of the other products that are on the market today, including Chorus CAD, which I helped develop on the basis of gene expression. So, I think the title of the session was Future Medicine, the Promise and Peril of Digital Medicine. And so to conclude, I will say that I believe that multi-omics or panomics will change the way we discover and develop drugs, develop biomarkers, and will change the way we practice medicine if, and here's my personal experience, I suppose, as an entrepreneur over the past 5 years, if we can find a way to fund our way to get to where we need to get. And I think that my experience has been that it has not been the easiest thing to get this kind of approach funded. And why is that? If you look at traditional funding sources like the NIH, they don't understand the approach. When we go to them, they say, what's your hypothesis? Well, we don't have one by design, and that doesn't fit very well. Big Pharma has been very interesting conversations for us because what we've seen is that the career drug developers have been raised in the looking at Western blots kind of era, and they don't know what to do with heat maps. When we talked to the venture capital guys in 2016, 2017, they either fund EBITDA, revenue, or they fund a drug development program, but they do not fund the data set. They do not fund the drug discovery, the target discovery and validation component, and the private equity groups, of course, only fund revenue. So it's an interesting place to be because we believe this is very disruptive and transformational, but to actually get to where we need to get is probably going to be the challenge. So I would say that in this panomic approach, I think genomics is easy, proteomics is easy, transcriptomics is easy. Figuring out the economics, that's not so easy. So to conclude, the balance between health and disease is determined in the context of the environment along this DNA, RNA, protein continuum, and I am convinced that that is the way for discovery. I think this will truly change how we develop drugs and biomarkers, and as I illustrated in my case presentation when I started, I think it will change the way we practice medicine along with everybody in the room, creating devices and all those components of the care system, if we work together to find a way to get there. So thank you for having me.